Hi students, welcome to HSC Biology and Module 5, Heredity. This is a slight deviation, a little bit of an extension on our study of autosomal inheritance, and I just want to focus in this video on dihybrid crosses. Specifically, what we have to do is we have to interpret examples of autosomal inheritance. Now that's a very broad idea. So we looked in the previous video at examples of monohybrid crosses, and I just wanted to introduce this idea of dihybrid crosses to you just so you've seen an example, and we'll look at one or two of them in class as well. So specifically what we want to do here is kind of hit the higher level of understanding, because of course if you can understand the principles and the practice of solving genetic problems that involve two characteristics, then you should theoretically at least, be able to feel more comfortable in solving traits with just the one. So let's just quickly review this idea of autosomal inheritance. Firstly, we said that autosomal inheritance relates to genes on the autosomal chromosome. So basically that's the non-sex determining chromosomes. And in humans, that's X and Y. So all of the other chromosomes would be autosomal, and they are the ones that we're looking at or talking about when we talk about autosomal inheritance. Each chromosome carries several tens to hundreds, to in humans probably about 1,000 genes on each chromosome. So that's a lot of information. And obviously there's no way that we can solve problems involving this entire human genome. We have to kind of look at little bits and pieces and hope that the understanding that we have of the practices that we use will be sufficient for us to extrapolate our understandings for more complex problems. So the key is that when we solve genetic problems, we can examine more than one trait at a time. Now this builds exponentially in terms of degree of difficulty and challenge. So we're just going to add a dihybrid cross just to the examples that we looked at previously on monohybrid crosses, and we'll do that again in the context of Mendel's P-plant experiments. So we're going to look at dihybrid crosses that involve an examination of two different characteristics. When we look at Mendel's experiments, we know that he looked at a range of different types of characteristics of pea plants. So when we were studying monohybrid crosses, we were looking at just one characteristic. So we could do the inheritance pattern for yellow seeds compared to green seeds. We could also work out the pattern of monohybrid cross for round seeds and or wrinkled seeds. These are individual characteristics. When we put them together, we have two traits, and these two traits together, when we start analyzing them in terms of their genetic probabilities, then we're talking about a dihybrid cross, and you'll see the hybrid nature of the cross as we go through. So as we did for monohybrid crosses, you need to assign letters, and the letters don't matter, but the letters should hopefully give you some indication of what it is that you're looking for, particularly when you're trying to interpret genotypes into what those phenotypes, how they would be expressed. So if we say that the yellow is dominant, we use a capital Y to indicate yellow color dominant over green. And then if we say that round are dominant over wrinkled, then we can use that capital R as the dominant allele. So when the two occur together, we understand what that means. So now you see, instead of having two letters, we have four letters. Two for the trait of seed color and two for the trait of seed shape. Now, the important thing here is that when we're looking at the process of meiosis, we're modeling that idea of how the gametes form. We need to make sure that we have two pieces of information, not just one going into each of those gametes, but we also need to make sure that we have one piece of information that relates to seed color and one piece of information that relates to seed shape. It's no good having a big Y, little y, and nothing to do with the R's. So this isn't going to work. So what we have to do is look at all the possible combinations that we could have. So given that there's two different alleles for color and two different alleles for shape, that means we've got 2 times 2, which is equal to 4 combinations. So in the gametes of pea plants, you would have these different combinations: a big Y with a big R, a big Y with a little r, a little y with a big R, and a little y with a little r. Now that's all the possible combinations. That's not all the possible combinations for every individual. So in order to find out the combinations of the individuals, we actually need to go through an example. So let's use an example following the method that Mendel did. So Mendel started with pure breeding plants that were homozygous. And what he wanted to do was to look at pure breeding for the dominant traits and pure breeding for the recessive traits, which means in our first example, capital Y, capital Y, capital R would be yellow, round seeds, and little y, little y, little r would be wrinkled and green seeds. So that means that when these two combinations of individuals are cross-fertilized, that the first one is only going to be able to, remember that in our gametes, we have to have one piece of information about the color and one piece of information about shape, and therefore there is only one combination from a pure breeding plant for those two traits. So that's capital Y, capital R. Likewise, for our pure breeding recessive plant, there's only one combination, and that is little y and little r, which means that when you cross these together, if you have again, remember that we're doing these as like mathematical multiplication tables, there's only one option, and that option is big Y, big R, little r, which means that all of the F1 individuals are going to have the genotype big Y, big R, little r. What does that mean? What do they look like? Well, hopefully, while I've been scribbling that, you've been thinking about that. And you know that because the big Y is dominant over the little y, that they're going to be yellow. And the big R is dominant over the little r, which means they're also going to be round. So that is our phenotype. So the P's that are produced from this particular cross are all yellow and round. But what about the next generation? What happens if we cross if we make a cross for our F2 generation from just individuals in that F1 generation? Well, this is where it gets a little bit more complicated. So the first thing we need to do is we need to remind ourselves that our parents are all big Y, big R, little r. And because of that, the big Y, big R, little r means that we can have two pieces of information potentially going in about color and two pieces of information going in about shape. That means that when we put these combinations together, given that we're talking about randomness, random segregation and independent assortment of these genes, that means that the gametes could be big Y, big R, big Y, little r, little y, big R, and little y, little r. Now that's for both the male and the female. So that means when we're doing our Punnett square, so when we get to our Punnett square needs to reflect all of these possible combinations. And now we have 4 combinations for one parent and 4 combinations for another parent, which means we're actually going to have 16 different combinations that are possible. Remember, this is probability. This isn't guaranteed. This is just giving you a bit of an idea of ratios. Now, again, like we did before, but obviously on a larger scale, you're going to multiply everything in these cells in order to get the full complement of all of these. And the problem is when you look at the genotypes, you actually have a very large number of genotypes. So I can see a big R, a big R, a big Y, and a big Y. That's the one that's right up there in the corner, and there's only one of those. Also, down in the bottom right corner is little r, little y, and there's only one of those as well. So we've got one of those to one of those. I can also see kind of going through this diagonal here that there are 4 lots of the big R, big Y, little y. So there's quite a few of those different examples from the crosses that could be produced in this particular example. But there's also a lot of others as well. And rather than going through each of these individually, you can see from this Punnett square that I've included all of the different genotypes that are there. And that's one of the things that makes these problems a little bit more involved, because you've got to write all 16 of the combinations down and obviously group them together, because you're not going to have 16 different ones. You're going to have some like these, which have been repeated and which you're going to have to group together in order to get the numbers. You can kind of see a little bit of a pattern happening in terms of trying to group these together, which is something that you can look at as you go through and work through an example yourself. The thing that we have to do, of course, is we have to turn all these different genotypes into phenotypes. And hopefully you've realized if we've got all of these different combinations, then we're going to get all 4 of the potential combinations. So we're going to get yellow and round. We're going to get yellow and wrinkled. We're going to get green and round. And we're going to get green and wrinkled. Now, I've given you this example because it actually shows the colors and the shapes with a little bit more ease, so you can kind of identify this. And what you'll notice is that we've got 1, 2, 3, 4, 5, 6, 7, 8, 9 of the yellow and round. You can see here nice round but green options. So they're round and green. Here we've got a little section that are wrinkled and yellow. So there's 3 of those there. And then just one solitary one at the bottom, which is the 2 recessive traits, green and wrinkled. So our phenotypic ratio is then 9 to 3 to 3 to 1. And you might remember on the last video, this is what I said we got when we looked at dihybrid crosses. Now remember, this is the hybrid because it's the combination. It's a heterozygous. So we're actually combining 2 heterozygous individuals here. We started with pure breeds, but when we do 2 heterozygous, we've got 2 hybrids, 2 that have brought these different alleles together, and that produces this nice Mendelian ratio of 9 to 3 to 3 to 1. So now we can just review these Mendelian ratios again. For monohybrid crosses, our genotype ratios are 1 to 2 to 1, and our phenotypic ratios are 3 to 1. For the dihybrid crosses, the genotypic ratios are much more complex, 1 to 2 to 2 to 4 to 2 to 2 to 1, that sort of thing. But we simplify that down into 9, which is the 2 dominant traits, to 3, 1 dominant, 1 recessive, to 3, the other dominant and other recessive, to 1, the 2 recessive traits. These are complex and they're worth going through, but have a look at an example just to make sure you feel comfortable in case you get a question about dihybrid crosses that you know how to answer it. And thanks for watching.